This brief video will highlight some features of the TranspoScope tool, which allows detailed visualization of transposon insertions into the host genome, predicted by a deep sequencing method called TIPseq. In this case, we're looking at insertions of human line 1 elements. The input to the viewer is the output of TIP-Seq Hunter, described in the accompanying paper by Tang et al. In this case, the insertion is on the bottom strand, and the viewer is focused on the genome line 1 junction demarcated by the blue line. Thus, the insertion is conceptually separated into two bins, the genome bin to the left of the blue line and the insertion bin to the right. Here we're looking at a pre-existing homozygous insertion present in all human cells. Let's drag the sequence to the left and have a closer look. The four tracks show two different types of read pairs, since the data comes from paired end sequencing. Track 1, or GG, in this case, both reads align to genomic segments flanking the transposon. In track 2, GL, one read is in the genomic region and one read is in the line 1 element. And track 3J, these are paired end reads, which include a read crossing the line 1 genome junction. And in track 4, all, you have all possible read pair combinations, including junction reads and other single-ended reads not shown in the tracks above. One of the great features of this viewer is that you can zoom in and show the individual bases in the reference sequence. So let's have a look. The reddish vertical lines indicate the nearest cut sites for the enzymes used in the TIP-Seq protocol. These enzymes do not cut the transposon segment at all and cut relatively rarely in the genome. The base pair consistency statistics show when you hover over the histogram. This data set looks excellent. However, consistency is unsurprisingly substantially lower in the poly A tail region. Homopolymer regions do not sequence well. Here's another insertion. This is a somatic insertion into the BRCA1 gene. The number of informative reads here is small, presumably because this somatic insertion is not present in 100% of the cells in the tumor sample. Only 3 junction reads can be viewed for BRCA1. So let's take a look at how they line up in the junction read viewer. The table on the right displays gene information. The color indicates whether the insertion site is located in an intergenic or intragenic region, with hot colors indicating the insertions most likely to affect a gene. So red would mean an exonic insertion, orange would mean an intronic insertion, and green or blue are intergenic insertions, with green closer to a gene and blue further away from a known gene. So you can see BRCA1 insertion is in an intron. You can also click on the link next to any insertion and visualize it on the UCSC browser. So let's have a look. Here, the insertion site for BRCA1 is represented by the little rectangle in the track. One more thing you can have a look at is the parameters for any TIPseq experiment. There's a lot of information about each experiment here. The plot shows 5 of the parameters used by TIPseq Hunter's machine learning algorithm. Evaluation of these parameters can help improve the sensitivity and specificity of the program to distinguish true insertions from noise. The 5 parameters are shown in the X and Y coordinates: the fill color, the outline color, and the size of the points. In the left column, you'll see the negative instances. These are the locations with no appropriate line 1 priming site. The middle column are the positive instances, or the locations of known insertions many of which will be present in any human sample. And the right column are the unlabeled, which is the rest of the locations. This will include the unknowns that are typically being sought from one of these experiments, the new insertions. So specific information about each location can be seen by hovering over each point. All of the parameters can be used to filter and see specific locations in more detail. You can use the sliders to zoom in or out as needed. The plot is customizable also and allows you to choose your own parameters for the X, Y, fill, outline, and size variables. Once you've decided on the variables, click Update to replot the graph. For example, let's change X to read depth and Y to probability that the insertion is real. Now we have a nice plot showing the probability as a function of read depth. Have fun using the TranspoScope to find some really interesting insertions in your data.